Welcome everyone. My name is Anup Mahulkar, and I lead the technical team at NeMO, where we implement the ingest and data dissemination portals. So I'm going to give a quick overview about the ecosystem, how it's configured, what we've accomplished over the last year, and what our plans are for the coming year. So this is sort of an overall architecture diagram of the Scott consortium. So essentially, most of the work we do is in the cloud. We have also a local replica of it, but I'm just presenting it from a cloud perspective to make it easier. So the idea is that we have different data storages. We have some data which is stored pure, the controlled access data is stored separately from the open access data store. And the idea is that we would be submitting the controlled access data storage to dbGaP, whereas the open access data will be made available through different mechanisms to our users. On the next slide, we sort of talk a little bit about the data flow. And so currently, people submit files to us through our ingest system. It's either a website or an API, and I'll talk a little bit about that. And the way you can access the information is that this is coming in the future. They're called landing pages, and I'll be talking about that. Or you can use HTTP server to go and get our files. Or we have a data discovery portal, and we also have a web-based gene expression tool called NeMO Analytics. And so there are different ways by which people can access information that is available at NeMO, and all of these will be made available over the coming year to the Scotch Consortium. So just to walk you through what happens when data is submitted to us. So we essentially treat data as three different buckets. So we have what we consider as public data, and there's embargoed data, and then there's what we consider as restricted data coming into us. And depending on where it comes, essentially we process it slightly differently. The first thing we do is we issue every asset that comes into NeMO is given a unique identifier. And that way, then people can refer to our files using that identifier, and people can retrieve it using that identifier. Then we process the data, and then we send it to the restricted release for restricted data. And within restricted release, there could be some summary data that's considered for public release, and that's moved into our public release area, and people can access that information from our tools. Now, for data are embargoed, then essentially they're treated differently. We'll keep them in a holding area till the embargo release time, and at that point, either data goes into a restricted release if it's controlled access, or it goes into a public release area from where people can access the data. And then finally, we have what we consider as the public or open access data that's processed and sent to our public resources immediately. As I mentioned, there are multiple ways of accessing the data. Currently, there's a data portal from where you can access it. There's NeMO Analytics, or if you want to analyze the data, you can go to the Terra Broad platform where you can access the data and manipulate it and analyze it. And there'll be presentations after mine which talk about these two aspects of using data on the Terra platform. And then there's a pilot that's going on where some of the data from NeMO will eventually be made also available on the Broad single cell portal. And so some Scotch data could also end up being on the Broad single cell portal if people are interested to sort of see their data in the Broad single cell portal. But that's something that doesn't exist. That's something that we're working on for the future. Now, when people submit us data, as I said, there are two ways of submitting data to us. One is either through a website, or we also have an application programming interface or API for people who want to submit to us programmatically. The submission to us is based on this concept of manifest files. So essentially, each submission includes a set of information about the files that are being sent to us. And that's how we track information. And each of our manifests gets assigned this unique identifier. And when you submit it, it's sort of either restricted or it's open access. That's what's indicated. And we have a dashboard that people can log into to see what is the status of a particular submission through each of the steps that we have. So whether the manifest is validated, whether the upload was complete, whether the QC was complete, and then where are we in the process of releasing the data to the consortium. And just to remind people, as of now, most of the data is being held within the consortium. It's not being released publicly. There may be a couple of studies which are at the point where we'll be sharing them publicly, but otherwise most of the data is currently available only to the consortium through all of these mechanisms. And so here's the dashboard, and the same information about the status can also be retrieved using an API, and we call it as a submission API. So people can use the submission API to know exactly what state is their submission in terms of each of these steps that I've outlined here so far. So just to give you an idea, over the last few years, these are the sort of project start dates and the amount of data that we have ingested into the Scorch portal for, at NeMO. So we have almost close to 10,000 individual files that have been sent to us, and it amounts to close to about 50 terabytes of data that's currently at the Scorch NeMO portal that we're holding for the consortium. And I talked about what's coming in terms of landing pages. So we're in the final phases of testing these landing pages. And the idea with the landing pages is that, as I mentioned before, each file or each asset, and the reason I use the term asset is because if you think about a subject or a grant, that's not really a file. So that we consider as an asset. So each asset has its own landing page. And that's one of the components that's required for meeting the fairness of a data repository, and that's where you're trying to get towards fairness. So each asset will have a landing page, and the landing page can be accessed using the unique identifiers that we assign to our assets. And the NeMO identifiers are registered with identifiers.org. So essentially, search engines index them. And so somebody were to search a particular identifier in the Google search engine, you should be able to pull up that particular asset using search engines, and essentially provides the basic information. So currently we have landing pages for, these are the sort of individual assets that we track within NeMO. So we track a grant, and within grant you could have different projects or studies. And there could be a publication that ensues from that, and that's also tracked independently. And then we have subject, a sample, a file, and a collection. So these are all the entities within NeMO that are tracked and that you can actually pull some information about it. So next few pages, I'm just going to give you a couple of examples of what those landing pages look like. So this is, for example, the landing page for a grant. So essentially, if this is a particular grant ID that we've assigned to it, NeMO GR and this particular cryptic code. So if you type that in, it pulls up this page and it gives the basic information about the grant. If there are any particular collections that are associated with the grant, then it gives you a list of the collections and the labs that are associated with the grant and the contributors and their ORCID IDs, and some documentation about how to cite it and how to sort of access information about this collection. So this is what a grant landing page looks like. And similarly, you can recreate collections, or users can request collections to be created. So collection is just a collection of set of files that you want to sort of tag and release as a bundle so people can reuse it. And typically what we use collections for is pages, for publications. So when you have a publication, we create a collection associated with the publication so that people can download all the data that's associated with that particular publication. So each collection again gets assigned a unique ID, and there's information about collections, and you can sort of information about how to access it, whether it's all the files, or whether you just want the raw files in the collection, or it could be the process files that are in the collection. So there's different mechanisms by which you can sort of look at these landing pages and navigate to them to download the data. And so here's this example of a subject landing page. So it gives some basic subject observations that we have and any taxonomies, and if there are any files associated with them, then those will also be listed in the future for that particular subject. And so here's an example for a sample and one for a file. So essentially, all of these are collections that will be released sometime in the Q1 of 2024. So people should be able to sort of get information about assets at NeMO through these landing pages. The switching gears now, the other way that people can access data at NeMO Scorch is through our data portal. And so the data portal at the top shows a summary of the current studies that are available at the Scorch portal. And most people should have access to it, and but you do need a login to access this data portal for the consortium. If not, please get in touch with Joe or one of us, and we can make sure that you have access to this portal. And so this portal essentially supports a faceted interface, and I'm just showing an example of the different facets we have. For instance, we can search by grants, we can filter your data by the organism of interest, or the viral status, or the brain region, or some of the techniques that are used for sort of narrowing down the files. So one can essentially use these faceted searches to narrow down the files that you want to work with or download, and then put them in a shopping cart and do something with them through the shopping cart. As I mentioned, the other tool we have deployed is called NeMO Analytics. And so this is essentially a tool that was built in collaboration with Dr. Rona Hertzano, who's a surgeon scientist. And this tool allows you to compare one or more gene expression or epigenetic data sets. So I'm giving you just an example of a single cell data set that's been shared through this Plaza Jennings et al. Paper that's available at NeMO Analytics. And so you can essentially look at either a single data set or you can create a profile with multiple data sets that are related. And so you can look at the gene expression for one or multiple genes through various displays through NeMO Analytics. And that's sort of one of the tools that we have deployed, and we're slowly sort of making some of the data available through the NeMO Analytics webpage. Okay. So switching gears to talk about some of the things that we have done over the last year. As you are all aware that we worked with you to finalize the Scotch manifest formats, which is how people will be submitting data to us in the future. There are two kinds of manifests, one's for human data and one's for non-human data. And so these have been finalized, and the CVs that we use with these metadata have also been finalized. And we have obtained the clinical data for most of the human subjects that will be in the data sets, and we are harmonizing the metadata across the brain banks to see how to sort of make facets available so you can search across data from different brain banks. And we also currently have consensus processing for over 100 samples that we are currently hosting at Scotch. From purely a technical standpoint or an engineering standpoint, what we have done is we have deployed the data portal for internal consortium use. Over the last year, we have designed a new database to accommodate collection and individual asset level tracking and some other information that was not being tracked beforehand. And we have implemented this new ingest system in the final week of stages of testing, and we'll be making it publicly available in Q1 of next year, sometime in the January-February timeframe. And as I mentioned, we have the landing pages that I showed, but the same information can also be accessed through an API if people are interested in accessing it through an API. And that should also be coming in the Q1 of 2024. So here's what to expect. We'll have an updated data portal to reflect the new database and the harmonized metadata. And we'll be doing a public release of both the data and the data portal. And what I mean by public release is this would be for people outside the SCOT consortium. So the idea is sometime early next year, we will make the data sets that are out of the embargo period will be made available publicly to the scientific community through the data portal that I've just displayed for you. Whereas the data that is still under embargo will be held back. The summary file will be made available to the archive, and then the restricted data will be going to dbGaP. So we will not make the restricted data available through the NeMO website. For people to access that, they'll have to go through dbGaP. And as I mentioned, some non-human data is already, for a couple of projects, is already available through the NeMO archive as of today. And there will be a data analysis and tools workshop in spring of 2024. And I something got covered here. The consensus process version of all the Scotch sequence data will be available, and we'll also make file-level tracking dashboard sometime in the coming year. So, and I believe that's all I have.
Yep. Before we swap over, we have a question in the chat from Iran. So Iran's asking, can you explain what consensus processing means in the context of us here?
Do you want to take that, Joe, or do you want me to take that?
If you want it, it's all yours. If not, I can take it.
Yeah, so I believe the consensus processing here means that we've essentially identified common pipelines that will be exercised across all the data that's at Scotch through the Terra platform and made available to the community.
Yep. So individual groups are free to run pipelines as they see fit, and then they can be uploaded to us as well. But the consensus processing in this context means we're going to be taking all the data and then running it in a way that we have well benchmarked, and so we can make it available as just another resource out to the community. And we have a couple more in the chat. Would you like me to read them out, Anoop? Or
yeah, I don't seem to be able to see it. So
okay, So we have one, are you planning to utilize AI applications in near future for data mining and analysis?
Not in the immediate future, but in the longer term, we are exploring different AI applications for running against the data that we're holding as card, but nothing in the next year or so.
Okay. And then we have one from asking, are the data files linked in any way to resulting publications?
Yes, and that's where I was talking about this concept of collections. So we'll essentially create collections which group all the files that are in a publication together, so people can identify and download the data associated with each of the publications.
Okay. And then one more asking, where can we view data once it's public?
So the faceted search portal that I displayed would be one place from where you can download the data. The other place would be that we essentially would have a HTTPS web-based data browser from where you can download the data from there as well.
And that will be for summary files as well, with restricted files having the extra step of working with dbGaP as well.
And I guess there's a question about when can we view it. So do you want to sort of talk about when the embargo period ends for different data sets?
Sure. So for all the data that comes to, it's generated in Scorch, it comes to the NeMO archive or the NeMO portal. And then from that date, we have a 12-month embargo period for internal members. And then once that 12 months hits, it becomes available out to the public. So if in the case of non-human data or model organism, that process is fairly straightforward. In the case of restricted human data, there are a couple more steps in that in terms of getting approval and move forward with that. But once it hits that 12 months, then it's open for people to either get access to the data directly or apply for access through dbGaP.
So we're about to move on, and let me just mention that we're doing very well with time. So the next two presenters, you're welcome to go until the break, which begins at 1:15.
Awesome, thanks. I'm just gonna start sharing my screen. Can you see my screen?
Yep, it looks great.
Thank you. My name is Asil. Today, we're going to be talking about cloud native pipelines available in Scorch. So our goal is to create open source cloud-based scalable workflows that meet analysis requirements for the Scorch consortium. So that means we want to make sure that our pipelines are fair. That's findable, accessible, interruptible, and reusable. And there are several features of such pipelines, such as being publicly available, being portable, written in workflow description language or WDL, being tested, documented, and modular. So to make our pipelines fair, we do this through partnerships. So we partner with consortium members who provide the code and tools in the cloud and into the cloud, and they validate their code. And what we do is we improve the performance of the code in the cloud, and we also provide documentation, testing, and results. So let's take a look at the upcoming and available pipelines. So what you see here is a timeline in green at the top, and we have the list of pipelines on the right. So we have the multi-omic, the single cell RNA, single cell attack, and single nucleus methylome. We notice that all these pipelines are available to use in the cloud today, and we are working on improving the performance and optimizing all these pipelines. So let's more specifically look at the multi-omic pipeline. So this is the multi-omic pipeline overview. As you can see, it takes in the gene expression FASTQ and attack FASTQ as input, and the pipeline comprises of two parts. So we see the top part, which is highlighted in green. This is for the gene expression part and is also known as Optimus. And we have the bottom part here, which is highlighted in blue, and this is the attack portion of the pipeline. So let's dig deeper into the gene expression portion of the pipeline. So the gene expression portion of the pipeline, or Optimus, takes as input gene expression paired FASTQ files. It takes in one library at a time. These FASTQ files are split into smaller FASTQs depending on barcode, and these smaller FASTQs are then analyzed in parallel using StarSolo, which maps the reads to a reference genome and also corrects for barcodes. This produces smaller BAMs, which are then merged together to give you one overall aligned BAM, and also gives you a count by gene matrix in H5AD format. From the aligned BAM, we also compute QC metrics in CSV format, which are also added to the H5AD file. So this concludes the gene expression portion. So let's take a look at the attack portion of the pipeline. Again, for the attack portion, we take in the attack FASTQ files as input. These FASTQ files are as similar to what we saw before, are split into smaller FASTQs. But in addition here, we also correct for barcodes. These smaller FASTQs are then analyzed in parallel, where we use cut adapt to trim the adapters, and then BWMM2 to align the trimmed reads to the reference genome. This produces smaller BAMs, which are then merged together to give you one overall aligned BAM. And then from this one overall aligned BAM, we use SnapAttack2, which was developed by Kai Zhang, to compute our fragment file. And from this fragment file, we compute the metrics and QC. So now that you've seen the overview of the multi-omic pipeline, let's see how we validated the pipeline. So here we're going to look at validating human gene expression data. And these plots were produced by Nelson Johnson from the Allen Institute. The x-axis for these plots correspond to Allen Institute's 10x cell ranger output. And the y-axis for both of these plots corresponds to the output produced by our pipeline. Each point here corresponds to a gene. Now let's take a look at the plot on the left. We notice that there are some genes called by Cell Ranger and not by Optimus. And the plots here were produced by the older version of StarSolo and prior to any optimization efforts. However, after collaborating with our consortium members and including new features of StarSolo, we produced this plot here where we notice that there are no genes not called by Optimus or Cell Ranger. So we see a tighter correlation between the two. Now that we validated our gene expression data, let's look at how we validated our attack portion of the data. As you see here, the x-axis for both corresponds again to this output by Cell Ranger, and the y-axis corresponds to the output by our pipeline. Each point corresponds to a barcode. So let's take a look at the plot on the left. This looks at the number of fragments per barcode, where we see a tight correlation between the outputs produced by our pipeline and that by Cell Ranger. After filtering those with a smaller number of fragments, we also look at the TSSE, which is the plot on the right, which is a transcription site start site enrichment per barcode, and we also see a tight correlation between the two here. Now that you've seen a little bit about the pipelines, I'm going to pass this on to Liz, who's going to go over Terra, which is our cloud-based bioinformatics platform to analyze this data. So I'm going to stop sharing my screen now.
Okay. Thank you so much, Asil. Let me share my screen. Okay. Okay. Can folks see this okay?
Yeah, before we get there, we have one question in the chat that may be better to hit now instead of wait till the end. So
Great.
we have a question. After the newest Optimus version, the only points away from the diagonal seem to have higher values for Optimus than for Cell Ranger. Is that correct? And do you have any idea of why that might be occurring?
Asil, you're on mute.
I was just saying, let me pull up the slides again. Give me a sec, sorry. So
You're visible.
we're talking about this plot here, right?
Yeah, I believe so.
Yes. So the question is there only points away from the diagonal seed to have higher for Optimus? Yes, that's correct, than Cell Ranger. do we understand why? I don't have an explanation as to why now. Unless someone else wants to pitch in.
Does anyone else, we have time, does anyone else on the call want to kick out some ideas?
One second, Hagen, I'm gonna allow you to talk, so feel free to either talk or drop in the chat, whatever's easier.
Yeah, well, I naively would look into gene families, genes with repetitive sequence, these kind of things. Yeah.
Great, thanks.
Okay, are we set to switch over to the next session, Joe?
Yeah, we can swap over. I don't see anything else in the chat, but thank you for those who have been dropping questions.
Thank you. And actually, I'm going to hand this over to Kaylee, who's going to be giving this presentation. Kaylee, are you able to share screen? Is she a panelist? There we go. Okay.
There we go.
Thank you.
Okay, it looks like my video, I can't start my video. I might need permissions to do that. Oh, there we go.
Should be right there. It just takes a
Perfect.
second.
Thank you. Okay. Let me go ahead and share my screen here. Okay, I think we're ready to go. So before we actually start talking about Terra, I first wanted to take a little bit of time to talk about the cloud and its advantages. So before the cloud became so popular, many people used what we now call the traditional approach to bioinformatics, where data lives in a central repository, and each researcher can make a copy of the data on their local machines. So with this approach, we're bringing the data to the researchers. This particular approach isn't ideal for a number of reasons, including that it can be costly, insecure, and it can lead to results that are not entirely reproducible. So instead, more and more researchers are turning to the cloud for this type of work, where data lives in a repository in the cloud, and researchers access the data instead of copying it. So with this approach, we aren't bringing the data to the researchers. Instead, we're bringing the researchers to the data, making the work more cost-effective, more secure, and more reproducible. This sounds great in theory, but clouds can be pretty difficult to use out of the box. There are a lot of things to set up, including options that probably aren't even relevant to the work that you're actually trying to do in the cloud. And that is where Terra comes in. It's designed to do the heavy lifting and help you move your analysis to the cloud. And it does that by being a hub in the cloud data ecosystem. It gives you the flexibility to find and access data, tools, computational resources, things of that nature, but it also allows you to bring those things to Terra and use them there as well. Terra allows you to access everything you need in one centralized place with these core capabilities that are listed here. So Terra has a data library where you can access public and controlled data sets. You can perform workflows in Terra, which allows you to perform analyses at scale using scripts. Terra has the ability to do interactive analysis. So you can think about analyzing data with built-in applications that you might already be familiar with, like Jupyter Notebooks, RStudio, Galaxy, that will allow you to analyze your data in real time interactively. And Terra also has these workspaces or shareable units where you can bring together data and tools so you can easily share those things with collaborators. So earlier, I just talked about accessing data sets. And earlier, Anoop provided a nice overview of NeMO. And the NeMO archive and Terra are actually integrated. So if you were to go to the NeMO archive, you could search for data that you're interested in. In NeMO, you can use filters or faceted searches to narrow down the data that you're looking at. You can select that data that you're interested in, and then you can actually export it to Terra. And this is just an example of what that might look like. But once I pull up an example workspace, I'll show you exactly where that data table would get imported into if you were to use this system. Okay, so excuse me. This workspace that I'm going to show you now is set up for testing and use of the multi-ohm pipeline, which Asil was just telling you about. So I'm going to go ahead and open this workspace and show you around the workspace a little bit. Okay, so this is our multi-ohm workspace. This is a public workspace, and this is the page that you'll land on when you open a workspace in Terra. It's called the dashboard. You'll find important information here about the workspace itself. This one in particular is telling us about the multi-ohm pipeline because that's what's showcased in this workspace. So there's a lot of information on this dashboard about the multi-ohm pipeline. You can see information about what it does and links to documentation for the pipeline if you're interested in learning more. It tells you about how you can run the workflow yourself in this example workspace using example data that we have preloaded in the workspace. It'll tell you about the workspace or the workflow inputs, optional inputs here, outputs that are produced by the pipeline. We have a section here for estimates for time and cost to run on the sample data that's provided in the workspace, and then just some additional information about the workspace and the workflow as well. Scrolling back up here, on the right side of our dashboard, we have some other important information like when the workspace was created, your access level, who owns the workspace, as well as the bucket name for the storage for the workspace. Okay, now I want to go to the next page here, this data page. This data page is a place where you can organize all of your data in your Terra workspace. The data is primarily organized into data tables, shown here on the left. And this is where the data that you export from NeMO will end up. It'll be in a data table like this, and that data table will look similar to the ones that are shown here. So each of these tables basically is a spreadsheet that allows you to input information about each of your samples, and then you can, outputs of workflows can be written to this table as well. And the cool thing about these tables is that in Terra, you can actually just pull this information directly from these tables into the analyses and the workflows that you can set up in these subsequent tabs here. Moving on to those, the next one here is the analyses page. This is a place where you can launch different files for applications like the ones that are shown here. As you can see, this particular example workspace doesn't have any of those files already loaded into it, but this is where you could create or upload those if you wanted to do that. And we have a workflows page. This page is where you can find and configure workflows that are written in the workflow description language or WDL. In this example workspace, the multi-ohm workspace, you can see we have the multi-ohm pipeline here that you can click on. And once that loads, you'll see the workflow configuration page where you can select data from your data tables, like I was just talking about, to pull that information right into your workflow. You can specify inputs down below. You can also specify outputs. And you can even see in this tab, the script itself that you would run on the data if you were to launch a workflow like this. I also want to show you this job history page. This is where you can monitor the status of your WDL workflow and troubleshoot if there's a problem. You can also see we have a successful run here of the multi-ohm pipeline that you can take a look at if you wanted to see how we had configured it or anything like that. Okay, and the last thing that I wanted to show you is how you can actually find this workspace. So there are a couple options. You can click to get to the workspaces page. You can click on this big workspaces heading up here, or you can click on the main menu here. I'll click on that. and then you can click on workspaces in this menu. Okay, so this is going to bring you to the workspaces page. There's a lot of ways on this page, as you can see, that you can narrow down your search for specific workspaces. For this one in particular, I'm going to click on public because I know this is a public workspace. And I think probably the easiest way to find this workspace is to just go ahead and search Multi-omic, and you can see it's the only workspace, the only public Multi-omic workspace. And so that's this workspace that we were just looking at. Okay, now switching back over, that wraps up our presentation on NeMO and Terra for today. So thank you all for listening. And we also wanted to, of course, thank our many, many collaborators for helping us with this work now and in the past and in the future. It would absolutely not be possible without all of them. So thank you very much. And that wraps us up for today.
Thanks.
For our portion, I should say.
Yep. So we have a question in the chat from Norman. So very nice integrative approach with NeMO. Would cloud storage restrict the types of analysis that could be done to what is available on the cloud, or can you use your own code to analyze the data?
Yeah, that's a great question. You can use your own code. It just may need to be adapted to use cloud resources for, say, containers or scripts, yeah, or locations of files.
Okay. And I see Alejandra has her hand up, so I'm gonna do that. You can go ahead and ask your question.
Alejandra, you're on mute.
I'm sorry, I think
There
I
we go.
did something. I don't have any question.
Okay.
Yeah, I'm sorry.
Oh, no problem.
It was a mistake.
Okay. Would you like to comment a bit on how, so these tools are being built for Scorch and Bican and other projects. Would you like to comment at all on that they're tools that are for the wider community, and in the way that how we're thinking about building them along the way? It's not just for these projects. Is there anything related to that you would like to comment on?
Are you asking me, Joe?
Yes. Yeah.
Sure. Yeah. Yeah. So I think one thing that we're always thinking about and is really important is making these tools and platforms applicable to a wide audience of scientists and researchers and groups and anyone who's interested in looking at this stuff. We have a really big focus on open source and making open source tools and making our work fair. As Asil said, that's a really big priority for the teams here. Yeah.
Great.
Couple more questions here. Can you comment on the data size limitations using the Terra system? Now is when you usually use the term infinitely scalable.
Yes, in theory. I don't know if any of our engineers want to comment on that.
That the term infinite was recently used to answer this question. So yes, it's pretty infinite as long as you still pay for what you use. So infinite might take a bit, but yeah, you're not limited by your data size.
Yeah. So Iran has a great zinger, which is the integration is promising, but how our Terra workflow is going to be integrated with the data that's at dbGaP. Anoop, are you?
Yeah, I can take that. So my understanding is that the consortium agreement allows people within the consortium to be able to access the restricted data once all the ICs are in place. And Joe, correct me if I'm wrong there. So what we would do is essentially so NeMO has the restricted data in our GCP buckets because we do the processing. So what we would essentially do is allow all the consortium members will have permission to read from that data. So essentially this integration will allow you consortium members to be able to work even on the protected or controlled access data in Terra using the GCP buckets that we've set up. So that should not be a problem that I foresee.
And I think we could also add that one of the things that we're looking forward to is, even though the raw data may be restricted, generation of information derived from that is something that we can make available publicly in some cases.
Yeah, so that groups and the consortium as a whole have put a fair bit of work into aligning all of our institutional consents. So instead of having to go out and ask for consents for 12 different studies, we'll have it all be under a single consent for the data.
Yeah. So we have another one for the Terra team. One person is asking that they have cloud credits, and they're wondering if they have a chance to use them for using Terra freely.
Yeah, I can take that one. The answer is yes. Terra is free to use. The only thing that you pay for when you're using Terra are the cloud resources that you use inside of Terra. And you can use those Google Cloud credits for that. Yeah, it just requires kind of linking billing projects together, which is probably more detail. There's a lot more detail to that, but we have pretty good documentation on how to do that and how to use those free Google credits with Terra.
Any other questions from the group? Ron's got one. Can you see it, Anoop?
Yes, I can.
All right.
Yes, the answer is yes, we will make that available after the 12-month embargo period. Is that right, Joe?
Yes, that is correct. So we'll have the derived data as well. And like Aran mentioned, that the sequencing data will be part of our archive as well. But we're hoping that these consensus processing helps people out with gaining access to data. It just takes one more barrier away from people using the data.
Yeah, I just noticed that Iran said is more for comment than not a questions.
We don't mind hearing a little bit of praise. Are there any other questions? Here's one. There's a question to, I think, to the person who brought up the cloud credit question, which was if you could give them the link for obtaining free Google credits, that would be helpful.
I think I just sent it, but somebody check and see. It's under answered questions.
Yep. I think you just responded to that one. And
all right. Anything else that people would like to bring up?
Okay.
All right. Let me just take a moment to sing the praises of the team that we have here. Anoop has been leading a whole lot of the data management processes here at NeMO. Our partnership with the Terra team has been absolutely outstanding in the last year or so, and we're really looking forward to the new developments that they're planning on putting into the interface. And we do an awful lot of project management together. We've learned a lot from each other. I've learned a lot from the project management processes that are at Terra. So one of our experiences is that they always seem to be on time and under budget. And that looks like that's happened with this session as well. So instead of going all the way up till 1:15, it looks like we can end a little bit early. So we're going to take a break and resume back at 1/2 the hour. Is that correct, Joe?
That is correct. So we will be back at 1:30. So don't go away. We have plenty of scientific presentations coming up. But we'll take a little break and we get up, stretch some legs, and see you all in a few moments. If you have questions for our presenters from this morning, feel free to keep throwing them in, and then we will answer them along the way. We'll see you back in